starting now to make sure we record. So after each lecture, we will put a link for downloading the videos for each recording. So first, a little bit of housekeeping about the module. So there will be a total of 20 2-hour lectures during week 4, 5, 11, and 12. And there will be 4 workshops so far scheduled for on Thursdays afternoons, or I guess your evening time. The marks will be split of 20% of the coursework and then 80% for the exam. For the coursework,
teacher.
Yes.
Can you hear me?
Yes.
You didn't share the screen.
Yes, sorry. Yes, sorry. Thank you. Share, I want to share 2. Sorry, thank you. Yeah, thank you. Can you see now? Can you see now? Okay. So for the marks, for the coursework, there are 3 components. There'll be a group poster, which involves preparing the poster and uploading for marking. It'll be, we've divided the class of 6 of you and groups of 6. And then there'll be, for one of the third workshop, you'll do a very short presentation of the poster to the class. And on the last week, there'll be a problem-based question exercise online. We'll make it available, and then it's to help you prepare for the exam, the type of styles of questions. Okay, this will be how your coursework grade will be marked. For the final exam, it should be standard across your other year 3 modules. It will be 80% of the module mark, comprising of 20% MCQs, 40% problem-based questions, and 40% essay questions. So we will start this week with an introduction to endocrine physiology and biochemistry. We'll give you an overview of the hormones involved and how do you measure them. We'll give you a little bit of anatomy and function of the hypothalamus and pituitary glands, and that will be covered by my colleague Carlos. And then he'll talk about diseases of the pituitary gland. And then we will have a workshop on Thursday about talking about the poster. What we expect, what a good poster looks like. Next week, we'll talk about the thyroid gland and diseases associated with the thyroid gland, the adrenal medulla and its hormones, the adrenal cortex and its hormones, and then we'll talk about the essay writing portion of the exam and what a good essay will look like. And then when we come back in May, together, we'll finish covering the adrenal cortex. We'll talk about pancreas and peptide hormones, and then lead into diabetes, type 1 and type 2. We'll have the poster presentation from you at that point. And then week 12, we'll cover how to treat type 2 diabetes. We'll talk about reproductive endocrinology, both male and female. And then we'll have a revision workshop to prepare you for the exam. So the content we will draw from will be from these 3 different textbooks. You can use any of the 3, should be sufficient. Pick your favorite one for content and to support what we're lecturing on here. There's also, occasionally we'll put up some research papers on QM+, and some review papers on the topics, and then a couple websites in terms of the treatments and the order of treatments that are useful when we start talking about how to treat. So this is one in particular. It's very good. So for today, I would like if at the end of these series of lectures today you were able to define what the endocrine system is, describe the gross anatomy and functions of the endocrine system, compare the endocrine system to the nervous system, define what a hormone is, describe the different classes of hormones, give me examples, and explain with examples the regulation of the endocrine system. So how does each organ communicate with each other? What is the connection between the 2? So we'll first start with telling you what the endocrine system is and why it's important, why we have an entire module on it. We'll talk a little bit about the gross anatomy of the endocrine system. We'll compare the endocrine and the nervous systems. We'll define, classify, and characterize the hormones involved. And then we'll talk about regulation, which the regulation in the endocrine system is a key part of its function. Okay? So, first, what is the endocrine system and why is it important? So the endocrine system is what defines who we are. Gives us our gender identity, male, female. It is what is involved when measuring for pregnancy. It's what controls contraception. It's what controls our height. What controls our bone structures, our blood pressure, diabetes, obesity. These are all endocrine disorders. Aging process, endocrine disruptors from the environment. So a very important topic these days about the environment and plastics in the environment and air pollution. Those are all endocrine disruptors. Cancer, very much related to our endocrine system. We'll talk about different tumors of the endocrine, and that's the expertise of Carlos. And then measuring the endocrine system throughout the age and life course is a very important aspect of health and disease. And then obviously one of the popular things in the news is athletes that are cheating, and they often use hormones that are related to the endocrine system. So, what does the endocrine system look like? By definition, the endocrine system is a system of ductless glands which secrete chemical hormones, chemical messengers we call them, into the blood. These hormones are then carried in the blood and are able to act on cells possessing the appropriate receptors in the target tissues. And then some of the target tissues are located here. So many people forget that the skin is an important endocrine system. So the endocrine system, typically we start at the top, and it's pineal gland within the brain, hypothalamus, and the pituitary. Okay, and then next down you have the thyroid and parathyroid glands, the skin, as I mentioned, the thymus, the heart, the liver, the adrenal, and then all of these target tissues play an important aspect. Okay, and these are typically what we call primary endocrine organs, and then these typically are secondary organs on the right side. Importantly, many organs act on several different target tissues, and the endocrine glands, as you can see, are distributed throughout the body. So it's a major regulator of many different important systems within the body. Sure, I'm not advancing. Let's see here. Can you see my? There we go. Okay. So let's talk a little bit about the gross anatomy of the endocrine system. This is the pituitary gland. So it's situated just below the brain, and it's encased within the skull. It's sometimes called the master gland. It's made up of two sides, the infundulum, okay, and it which leads down into the posterior pituitary gland, and then you've got the anterior pituitary gland, which is fed in by the hypophyseal portal vessels. And at the top of the pituitary gland is the hypothalamus, feeding in these neurosecretory cells. So the neurosecretory cells feed into the posterior pituitary gland, and these hypophyseal portal vessel into the anterior pituitary gland. This is just a view of the connections between the surrounding CNS and sort of where the pituitary gland sits within the brain. Just above it are the optic chiasm, and below are the sinus cavity. You can see the pituitary, although very important, is actually quite small. The thyroid and parathyroid glands sit down below. These are the parathyroids just sitting on top here of the thyroid, these four small circles. They're situated in their neck and an important control of the metabolic rate and calcium homeostasis. We have the adrenal glands, which are down on top of your kidneys. The adrenal gland that you'll hear about is made up of two major areas: the adrenal cortex on the outside and the adrenal medulla on the inside. Typically, these two regions control, with the medulla controlling stress response and the cortex stress, sodium and glucose homeostasis. The pancreas is an important organ sitting just next to the small intestine, and it's a very important aspect for our exocrine function. So it secretes digestive enzymes directly into the GI tract. It helps break down food, and one of the major aspects of this is to secrete insulin. And this insulin, which we'll talk about, obviously impacts the ability to regulate our blood glucose concentrations and is a major player in terms of diabetes. If you were to open up the pancreas and have a look from a cross section, you can see the pancreas is made up of what are called islet cells, and then next to it you have these exocrine portions of the pancreas. But in terms of typically insulin production, we'll talk a lot about these islet cells here in blue. And then obviously we'll be interested in the difference between endocrine and exocrine glands. I mentioned to you an important part of the endocrine system is the reproductive system, and the two important organs involved are the ovaries or testes of the gonads, which control secretion of the male and female reproductive hormones. And these are responsible for the development of secondary sexual characteristics and reproduction. So let's compare the endocrine and the nervous systems. So if we have two cells that are required to communicate, what are the different options available to them? Okay, we've got cell A here, and we've got cell B. The simplest way that you would have learned about in your cell biology is that cell A releases a chemical substance to cell B, and we might call this a messenger of some kind. This chemical substance will then influence the activity of cell B, and cell B will respond by growing, by moving, by dying. So what is the major problem for this simple way of communicating? It's nonspecific, okay? So the chemical isn't just for cell B. It could be recognized by many other different cells. And a large amount of this message or chemical would be required for cell B to feel it, right? To experience it. And if the cells are far apart, it becomes obviously more tricky. The amount of substance that must be secreted thus increases, okay? Because they have to travel a long way, right? If we don't have a nice VOOV call, right? Then I have to be really, really loud for you to hear me all the way over in China. But we develop VOOV, we use VOOV so that my message can be directly transmitted from me to you, right? Well, the cell has a very similar system. And obviously, if this, if the distance creates a greater problem of non-specificity, right? We don't want my message to go all the way to everyone at the campus there, or we don't want all of Nanchang to hear Dr. McCormick talking about endocrinology. We don't want, Shanghai doesn't care, right? So how do we create specificity between London and the lecture theater there at the university? So one way is that the cells can release a chemical messenger and they can grow towards each other, right? So we can create a VOOV and I can send that VOOV and grow it all the way out specifically to your room. And so whatever I say only gets released, the message, my message, which we'll call the ni hao message. My ni hao message only gets released into your room there at the lecture theater, okay? And the cell develops the same idea. So the development of outgrowths from cell A towards the target B will then release what we'll call our ni hao message or ni hao compound and will be received by cell B explicitly. And this will allow A to then become further specialized by only releasing the chemical transmitter, okay, from one area of its surface. So cell A doesn't have to say ni hao everywhere. It can just say ni hao only at the end here towards the part that's connected with cell B. This type of specialization is the beginning of a nervous cell, okay? And it's how the nervous system operates. So here we have a series of two nerves. Cell A that's stretched along an axon and has very specific release at the end of these dendrites into cell B, this other neuron, for specific communication. The advantage of this system that the cell uses and the body uses is the nervous system allows very rapid, focused, precise communication, just like VUV, between individual cells. Okay? But what is the disadvantage of this system? So each cell can only communicate directly with a limited number of other cells. So how many people fit in the lecture theater there, right? Maybe 200. So we're limited in my ni hao message. My ni hao hormone is limited in the number of people I can communicate with this type of system, right? So the body requires an alternative method by which a single cell can communicate with a large number of widely distributed cells, right? So how am I going to talk to all the year 3 students or all of the biology students at Nanchang or all the medical students? So this is one of the reasons why the endocrine system was developed by the body. So an alternative system that the body uses is where cell A can release the chemical messenger into a transport system present within the organism, and that is the blood. So we release the message into the circulatory system, okay? And we can imagine it's the water system there at the university, right? So we release it into the water system, and we can have a message passed along the water system. You drink the water, and you receive the ni hao message. And this allows the same thing, our hormone released by A, and because B's on the other end of the blood, or our water system, it will receive the hormone. So this allows less chemical transmitter to be needed, and it can quickly reach a large number of cells. So instead of a targeted ni hao via VUV into a room of lecture theater, I can put the ni hao into the water. Everyone drinks water on campus, and everyone gets a nice ni hao message from London. And I only need to say it once. So less chemical transmitter needed to be released by A, and a large number of cells can be quickly reached easily. So what is the problem with this type of communication? One issue is you're limited. You have to be able to get the transmitter substance into the blood vessel, right? So it's got to be able to get in and it's got to be able to get out. So substances with a molecular weight above roughly 70 kilodaltons, they cannot leave the blood vessels. They cannot transport across the blood endothelial cell membranes, okay? In addition, transmitter substances, there's communication you'll remember, or if you'll learn certainly from Richard Gross when you do your tumor biology, but you've probably had some angiogenesis already, that substances can enter or leave the vascular system via typically the capillaries only. So the capillaries are the very small blood vessels that communicate with the cells. And this is where the exchange between cells and the blood system work. So you're limited in where you can enter your NIHAO hormone. Has to be either wherever the capillaries are. It can't just be wherever any blood vessel is located. So location is a bit of a problem, right? It's a bit like for your water system, if we use the analogy of the water system on campus, right? I put a NIHAO message into the water. You can only get the water where you have a tap, where you drink, or a drinking fountain. It's not going to be everywhere. So, what are some of the solutions that the body has developed to try to overcome some of these problems? One is all the cells producing the transmitter substance, they clump together to increase the amount of chemical substance release, okay? And this is developed into what are called glands. So, imagine there's many of me here in London, and we all sit there in one place to release our NIHAO hormone. So we only say one NIHAO, but there's about 50 of me, and we're all saying NIHAO at the same time, okay? And that would be the NIHAO gland. In addition, it was developed so that each transmitter is very potent, okay? And it targets a specific receptor on the target cell so that only a small amount of transmitter is required to bind to this receptor at the target tissue. So a small amount of NIHAO, and NIHAO is extremely potent. It puts a smile on everybody's face when it binds to the receptor. And everyone has that receptor. So the body developed the system of chemical transmitters that work at very low concentration and bind specifically to a given receptor only located on the target cell B. And this system has developed and evolved into what we call the endocrine system. And this allows very slow, sustained communication between a group of cells, a large group of cells. So it's not fast, but it can quickly get out a message across to a large number of people with just a small release of hormone. And it's sustained. Now, these two systems, the nervous system and the endocrine system, they don't work separately. They are very much linked together, and they work together in part because of what I just explained to you, that because the nervous system works very quickly, it can then trigger the endocrine system to now work more specifically. So imagine you send me a message very quickly via VOOV that says, hey, Peter, we need to make Nanchang campus very happy. Send out a nice, warm NIHAO message to everybody. So you would quickly be the nervous system, you would open up your VUV, you would send me a very quick message, and then I would go into the water in the endocrine system and release a nice message for the whole campus to say, hey, good morning, ni hao. And this is exactly how the nervous and the endocrine system work together, okay? The major location, they interact at several different places, but a major one is the hypothalamus. And here is where the two nervous system and endocrine system overlap and interact with each other to control body temperature, circadian rhythms, appetite, to name a few. So, just a visual cartoon on a reminder of the endocrine versus neuronal signaling, so you can have very clear in your mind. For the nervous system, we have a stimulus that comes into the neuron. This travels down an axon in a very fast, typically electrical manner, and is released at a very specific location, and that is transmitted all the way down until you get a response at a cell type, okay? For hormones, for the endocrine system, we have that general stimulus that comes into an endocrine cell, and then the hormone released into the blood vessels. This signal then travels everywhere within the bloodstream, okay? So it's not released in a specific place. Wherever you have those blood vessels go and the capillaries, you get that exchange with the capillaries to the cells, and then you have a specific receptor on the target cells, which bind that hormone and then lead to a response. Okay? So one of my favorite questions on exams is, how does the endocrine system compare or differ from the neural system? Now, just to zoom in and reminder that, because these will be one of our favorite things to talk about in this lecture, in these lectures, will be the receptors. So in the hypothalamus, you have two different types of endocrine release. You have what are called neurosecretory cells, which are a hybrid of endocrine cells and neural cells. So these are, they look like neurons, but they release hormones. Okay, so you got a neurosecretory cell that comes down, looks very much like a hormone. It's going to release a hormone into the blood vessel. Okay, so instead of another neuron here, right here I'm showing you, this is one neuron, two neurons. Here I'm showing you one neuron into the blood vessel. Okay, so the hypothalamus has these neurosecretory cells which look like neurons and then release them but into the blood vessel. And then these later travel down and hit another tissue, bind a receptor, and give a response. Okay? So that's the difference between a neurosecretory or neuroendocrine cell and a neuron. Here the neuron is coming down and it's releasing onto another neuron. So that's neural signaling, and this is neuroendocrine signaling. So the neuron, it looks very similar to a neuron to this part, but instead of releasing to another neuron, it releases straight to the blood vessel. Okay? And this is, the hypothalamus has these types of cells. Now, one of the major differences between the neuroendocrine signaling versus neural cell signaling is the speed. Okay, so timing and duration is a very important part that separates the neural system from the endocrine system. So neurotransmitter secretion, something like, say, dopamine, you've got this action potential that I mentioned to you, and then small molecule dopamine being released onto a target cell. In that neurohormone in the hypothalamus situation that I just talked about, you would have the same action potential, which is fast, but the actual hormone's released into the blood. And this transmission is much, much slower, okay? This part is slower. This part is pretty much the same. So that's the difference. If I ask you, what's the difference between neurotransmitter and neuroendocrine signaling? Again, one of the important aspects of the endocrine system are the feedback. Okay, so feedback and communication between the different cell types. So how is this achieved? This should be a reminder or a review for you for some of your cell biology, but we have what's called autocrine signaling. If you remember, a cell can release a hormone. That hormone can bind on a receptor on the same cell and then shut down or change and alter this secretion or change something else. That's called autocrine signaling. You also have paracrine signaling, where a hormone is released from a cell and then it binds a nearby cell, okay, and alters some aspect of that nearby cell physiology. Or you have typically what we call endocrine action, which is what I've been talking about today, which is this hormone is released into the circulation and then binds a receptor on a distant cell. So all of these types of signaling actions can occur within the endocrine system. But the major one that we typically think about is this endocrine action via the circulation. So just a quick review. This would be endocrine signaling, where hormone is released from a cell into the blood vessel goes out, releases those hormones and through the capillaries, and you get a response. So it occurs between distant cells. The hormone is transported by the circulatory system to the target cells. What about this system? This would be what we call paracrine signaling, which occurs between local cells, and the target cell is near the signal releasing cells. And this is an example of our autocrine signaling, where the hormone is released, binds to its proper cell, and it alters a response on its own cell. So again, you should be able to tell me and explain to me different types of signaling and how typically the endocrine signaling is different from paracrine and autocrine signaling. And if I put up to you a scheme of these different types of cells or different types of signaling and cell communication, you should be able to label them and tell me who's who and draw these in your answers. Again, one of my favorite questions on exams. So, we've talked about what the endocrine system is and why it's important. We've talked about the gross anatomy of the endocrine system. We've compared the endocrine system and the nervous systems. And now we will define, classify, and characterize some of the hormones, okay? So first, just quick history lesson on how hormones were identified. So they were identified by this gentleman, or he sort of, they had been proposed, let's put it that way, prior to him, but he was the first one to test experimentally and identify the chemical substance of what we call hormones today. And his name was Arnold Bortold, and he was alive in the 1800s, and he did the following experiment. So he took a normal male chicken, what we call a cockerel in English, and a normal hen, a female chicken. And for any of you that have ever been around chickens, it's obviously can be difficult to tell the difference between a cockerel and a hen when they're especially when they're young. But as they get older, it's obviously a bit more easy, but it obviously becomes important. So how do you tell the difference? So what he did was he took the following 3 experimental groups. He first took these two different male chickens, and he removed their testes, okay? Both testes removed. And then he looked to see what happened in the development of these chickens. And what he found was, well, obviously I wanted to tell you, you can tell the difference between, right? An adult male chicken has a very big comb and a very big wattle, and a female, very small and very small. Okay, that's the visual. That's how you know as an adult. Obviously, if you're a chicken farmer, you know this when they get big, but when they're little, you can't tell the difference. So he removed both testes of these young chickens, and then he measured the size of their combs and their wattles as they got older. And he noticed when both testes were removed that the comb and wattles were very small. These chickens had no interest in hens, okay, no interest in reproduction. They were very, they sounded very weak when they mowed cock-a-doodle-doo in the morning. It was very weak, and they never fought. You know, chickens can be very vicious and fight quite a bit. For any of you who ever been around chickens, I was terrified as a child of a chicken because they, especially the male, they can be quite aggressive. So he noticed this when both testes were removed. Then he removed only one testes, and so he replaced one of the testes. Okay, and in this case, he could see that the comb and the wattles were now normal, that they now were interested in the hens, and they had a normal crow, and were again aggressive, and that the testes were larger in this situation than in the controls, okay? And then he thought, okay, well, what if I exchange the 2 testes, okay? Is it specific to the gland, or is it something the gland is releasing, okay? And so when he exchanged the testes, okay, he got the same result as 2, okay? So this told him that it wasn't having the physical gland per se of that chicken, but it was something being released from the testes that clearly was making the difference between these behaviors. So some sort of substance that was released into the blood, not the actual gland itself, that was making the difference, okay? So he concluded that the testes exerted an action on the blood to influence male development and appearance and behavior. So how could the presence of a testes exert an action on the blood? And that was obviously the discovery of the hormones produced and made by the testes. Okay, so that was the first experiment for validating the idea of hormones and a chemical substance that was released by a part of the body into the blood. Okay, so now we'll take a 30-minute pause, and I'll let you relax for a bit, and then we will come back and finish off today's lecture.